Hi, in this video I'm going to discuss about the metabolism of the nucleotides. So let's get started. First question is the biosynthesis of the purine nucleotides. There are de novo synthesis and the salvage pathway. Okay, biosynthesis of the purine nucleotides. Many compounds contribute to the purine ring of the nucleotides. N1 of purine means this is derived from the amino group of the aspartate, and C2 and C8 arises from the formate of N10 formyl THF. C2 and C8 is derived from the formate N10 THF. Okay, and N3 and N9 are obtained from the amide group. N3 and N9 are obtained from the amide group of the which glutamine. C4, C5 and C7 are contributed by glycine. C4, C5 and C7, these are contributed by what? N7. These are contributed by the glycine. C6 come from C2, CO2. It should be CO2. Okay. Two ways of biosynthesis. These are de novo and the salvage pathway. De novo, formation of the purine nucleotide from simple acyclic precursor, mainly in the liver. Okay, formation of purine nucleotide from where? Simple acyclic precursor occur in the liver. And the salvage reservoir pathway, using of purine base formed in the decomposition of the nucleotides. Okay, purine base is formed in the decomposition of nucleotides in the out of the liver tissue. Where? Out of the liver tissue. Purine can be directly converted to the corresponding nucleotides like this. For example, adenine to adenosine monophosphate, guanine to guanosine monophosphate, and hypoxanthine to ionosine monophosphate. It is for the erythrocyte and brain where de novo not work. Okay, means salvage pathway is for the erythrocyte and brain where de novo will not work. Next question is about the regulation of the purine nucleotide biosynthesis and the feedback inhibition, mainly dependent on the ability of the ribose 5-phosphate and the enzyme PRPP synthesis. PRPP synthesis means phosphoribosyl pyrophosphate. Okay, first there we have ribose 5-phosphate, then it converted into phosphoribosyl phosphate means PRPP by the enzyme PRPP synthase, and that converted into 5-phosphoribosyl 1-amine, and that converted into inosine monophosphate. Then break into the adenosaccinate and xanthinate. Adenosaccinate make AMP, means adenosine monophosphate, and xanthinate make GMP. After that, the sufficient amount will make in the body, they make negative feedback here to the here, and the production of the GMP will become normal. Okay, and also give negative feedback to this. Okay, that's done. And the next question is the catabolism of the purine nucleotides. In metabolism there is also anabolism and catabolism. Catabolism means breaking, and first is there in the purine nucleotides. There are pyridinosyne monophosphate, guanosine monophosphate, and inosine monophosphate. They convert into adenosine, guanosine, and inosine. And amino group removed from AMP or adenosine form what? Inosine monophosphate and inosine. AMP, when amino group removed from the adenosine monophosphate, it forms IMP, and when amino group removed from the adenosine, it forms inosine. Then inosine make converted into hypoxanthine, and guanosine converted into guanine, and adenosine converted into inosine. Okay, because the enzyme not present to convert adenosine into adenine, that's why it's converted only into inosine. And guanine converted into xanthine, the xanthine, and the hypoxanthine also converted into the xanthine by the enzyme xanthine oxidase. End product is of this metabolism is the uric acid. Okay, done. And the next question is the disturbance of purine nucleotide metabolism. GOAT, Lesch-Nyssen syndrome, and immunodeficiency disorder. GOAT, it's a disease, inherited disease accompanied with hyperuricemia, means if uric acid will increase in the body, and crystallization of uric acid and its salt in joint. If accumulation of the uric acid salt in the joint and the cartilage and the kidney, then the GOAT disease we will see. Symptoms are the joint inflammation and the acute pain, okay, and renal stones, the kidney stones, and the tophusus, means it's occur in the soft tissues. Treatment by the allopurinol. Also the treatment of the Lesch-Nyssen syndrome is by the allopurinol. Okay, now Lesch-Nyssen syndrome, it's inherited disorder caused by the deficiency of the enzyme. Which enzyme? Hypoxanthine guanine phosphoribosyl transferase. And Lesch-Nyssen syndrome is present at birth in baby boy because of the X chromosome. It's situated in the X chromosome. That's why, due to the overproduction of the uric acid, this disease cause overproduction of the uric acid. So decrease the uric acid, we use allopurinol. Okay, and hypoxanthine and guanine are not used in the salvage pathway of purine nucleotide synthesis. Okay, hypoxanthine and guanine are not used in the salvage pathway of purine nucleotide synthesis. So hypoxanthine and guanine are not utilized repeatedly but converted into uric acid. Okay, they are converted into uric acid. Symptoms are the severe gout symptom, and severe mental and physical problems. Okay, and self-mutilating behavior, means biting of the finger and lips continuously. Treatment by the allopurinol that I earlier said. Okay, and the last disease is the immunodeficiency disease. To define immunodeficiency disorders, mainly the enzyme defects, the deficiency of adenosine deaminase, means the ADA, maybe you heard about it earlier, cause severe combined immunodeficiency SCID, which causes T and B cell dysfunction. Okay, and deficiency of the purine nucleotide phosphorylase PNP cause only T cells. Adenosine deaminase cause only T and B cells, and deficiency of this, and deficiency of purine nucleotide phosphorylase cause only T cell. Biosynthesis of purine nucleotide, pyrimidine nucleotide by de novo synthesis and the salvage pathway. It's much similar like the purine synthesis. Aspartate, glutamine, amino group, glutamine and CO2 contribute to the atom in the formation of the pyrimidine ring. Like this, this is the pyrimidine ring, and aspartate, from aspartate 3 carbons and 1 nitrogen are formed, and from glutamine 1 nitrogen, and from CO2 1 carbon. And the next question is about the regulation of pyrimidine nucleotide biosynthesis. Okay, when aspartate and carboamyl phosphate are there, then by the help of the enzyme aspartate trans carboamylase ATPase, and by ATPase, and with the ATP form with energy means form carb amylase aspartate, and then form uridine monophosphate, uridine triphosphate, and then into the cytosine triphosphate. After production of the sufficient amount of cytosine triphosphate, then it gives the negative feedback, and the amount of cytosine triphosphate will be in the normal. And the next question is the catabolism of the pyrimidine nucleotide. When there is pyrimidine nucleotide, dephosphorylation, deamination, and cleavage of glycosidic bond form the pyrimidine, means catabolize the pyrimidine nucleotide into nitrogenous base, which are the cytosine, uracil, and the thymine. After that, the nitrogenous base converted into B-alanine and B-amino isobutyrate. What? B-alanine and B-amino isobutyrate. Then by the process of transamination, it formed acetyl coenzyme and succinyl coenzyme. Okay, understand catabolism. What? Acetyl coenzyme, succinyl coenzyme. Then the next and the last question is about the disturbance of the pyrimidine nucleoside metabolism, which disease is the orotic aciduria. It's the inherited disorder of the pyrimidine synthesis caused by the deficiency of the symptoms of enzyme of orotate phosphoribosyltransferase and decarboxylase. Deficiency of these two enzymes cause this disease. Symptoms are the excess of the orotic acid and excretion with urine. Orotic acid will be increased and it excretes with the urine. Mental and physical deterioration there we will see, and megaloblastic anemia. Treatment is uridine. The patient should take uridine for lifetime. Okay, that's all.